Class 11 Chapter 6 Anatomy of Flowering Plants. You can very easily see the structural similarities and variation in the external morphology of the larger living organisms, both plants and animals. Similarly, if we were to study the internal structure, one also finds several similarities as well as difference. This chapter introduces you to the internal structure and functional organization of higher plants. A study of internal structure of plants is called anatomy. Plants have cells as the basic unit. Cells are organized into tissue, and in turn the tissues are organized into organs. Different organs in a plant show difference in their internal structure. Within angiosperm, the monocots and dicots are also seen to be anatomically different. Internal structure also show adaptation to diverse environments. 6.1 The tissues. A tissue is a group of cells having common origin and usually performing a common function. A plant is made of different kinds of tissue. Tissues are classified into two main groups, namely meristematic and permanent. Tissues are based on whether the cells being formed are capable of dividing or not. 6.1.1 Meristematic tissues. Growth in plant is largely restricted to a specialized region of active cell divisions called meristem. Plants have different kinds of meristems. The meristem which occur at the tip of roots and shoots and produce primary tissue are called apical meristem. Root apical meristem occupies the tip of roots while the shoot apical meristem occupies the distant most region of the stem axis. During the formation of leaves and elongation of stems, some cells left behind from shoot apical meristem constitute the axillary bud. Such buds are present in the axil of leaves and are capable of forming branch or flower. The meristem which occurs between the mature tissue known as intercalary meristem. They occur in grasses and regenerate part removed by the grazing herbivores. Both apical meristem and intercalary meristem are primary meristem because they appear early in life of plants and contribute to the formation of the primary plant body. The meristem that occurs in the mature regions of roots and shoots of the many plants, particularly those that produce woody axis and appears later than primary meristem, is called secondary or lateral meristem. They are cylindrical meristem, fascicular vascular cambium, interfascicular cambium, and cork cambium are examples of lateral meristem. These are responsible for producing the secondary tissue. Following division of cells in both primary as well as secondary meristem, the newly formed cell become structurally and functionally specialized and lose the ability to divide. Such cells are termed as permanent or mature cell and constitute the permanent tissue. During the formation of primary plant body, a specific region of the apical meristem produce dermal tissue, crown tissue, and vascular tissue. 6.1.2 Permanent tissues. The cells of the permanent tissue do not generally divide further. Permanent tissue having all cells similar in structure and function are called simple tissues. Permanent tissue having many different types of cells are called complex tissue. 6.1.2.1 Simple tissues. A simple tissue is made of only one type of cell. The various simple tissue in plants are parenchyma, collenchyma, and sclerenchyma. Parenchyma form the major component within organs. The cells of the parenchyma are generally isodiametric. They may be spherical, oval, round, polygonal, or elongated in shape. Their walls are thin and made up of cellulose. They may either be closely packed or have a small intercellular space. The parenchyma perform various functions like photosynthesis, storage, secretion. The collenchyma occur in layer below the epidermis in the most of the dicotyledonous plants. It is found either as a homogeneous layer or in patches. It consists of cells which are much thickened at the corner due to deposition of cellulose, hemicellulose, and pectin. Collenchymatous cells may be oval, spherical, or polygonal and often contain chloroplast. They cell assimilate food when they contain chloroplast. Intercellular space are absent. They provide mechanical support to the growing part of the plant such as young stem and petioles of a leaf. Sclerenchyma consists of long, narrow cells with thick and lignified cell walls having a few or numerous pits. They are usually dead and without protoplast. On the basis of the variation in the form, structure, origin, development, sclerenchyma may be either fiber or sclerites. The fibers are thick, walled, elongated, and pointed cell, generally occurring in groups in various parts of the plant. The sclerites are spherical, oval, or cylindrical, highly thickened dead cell with very narrow cavities, lumen. These are commonly found in the fruit wall of nuts, pulp of fruit like guava, pear, and sapota, seed coat of legume, and leaves of tea. Sclerenchyma provide mechanical support to organs. 6.1.2.2 Complex tissue. The complex tissue are made of more than one type of cells and these work together as a unit. Xylem and phloem constitute the complex tissue in plant. Xylem function as a conducting tissue for water and mineral from root to stem and leaves. It also provides mechanical strength to the plant parts. It is composed of four different kinds of elements, namely trachea, vessel, xylem fiber, and xylem parenchyma. Gymnosperm lack vessel in their xylem. Tracheae are elongated or tube-like cell with thick and lignified walls and tapering end. These are dead and are without protoplasm. The inner layer of the cell wall have thickening which vary in form. In flowering plant, tracheates and vessel are the main water transporting element. Vessel is the long cylindrical tube-like structure made of many cells called vessel member, each with lignified wall and large central cavity. The vessel cells are also devoid protoplasm. Vessel members are interconnected through a perforation in their common wall. The presence of vessel is the characteristic feature of angiosperm. Xylem fiber have highly thickened wall and obliterated central lumen. These may either be septate or aseptate. Xylem parenchyma cells are living and thin-walled, and their cell walls are made of cellulose. They store food material in the form of starch or fat and other substance like tannin. The radial conduction of water takes place by the ray parenchymatous cell. Primary xylem is two types, protoxylem and metaxylem. The first form primary xylem element are called protoxylem and later formed primary xylem is called metaxylem. In stem, the protoxylem lies toward the center pith and the metaxylem lies toward the periphery of the organ. This type of primary xylem is called indark. In root, protoxylem lies toward periphery and metaxylem lies toward the center. Such arrangement of the primary xylem is called exarch. Phloem transport food material usually from leaf to other part of the plant. Phloem in angiosperm is composed of sheep tube element, companion cell, phloem parenchyma, and phloem fibers. Gymnosperm have albuminous cell and sheep cell. They lack sheep tube and companion cell. Sheep tube elements are also long tube-like structure arranged longitudinally and are associated with the companion cells. Their end wall are perforated in a sieve-like manner to form the sieve plate. A mature sieve element possesses a periphery, cytoplasm, and a large vacuole but lack a nucleus. The function of sieve tubes are controlled by the nucleus of companion cell. The companion cell are a specialized parenchymatous cell. And phloem fibers. Gymnosperm have albuminous cells and sheep cells. They lack sheep tubes and companion cell. Sheep tube elements are also long, tube-like structure arranged longitudinally and are associated with the companion cell. Their end wall are perforated in a sieve-like manner to form the sieve plate. A mature sieve element possesses a peripheral, cytoplasm, and large vacuole but lack a nucleus. The function of sieve tubes are controlled by the nucleus of companion cell. The companion cells are specialized parenchymatous cells which are closely associated with the sieve tube elements. The sieve tube element and companion cell are connected by pith field present between their common longitudinal walls. The companion cell helps maintaining the pressure gradients in the sieve tube. Phloem parenchyma is made up of elongated tapering cylindrical cells which have dense cytoplasm and nucleus. The cell wall is composed of cellulose and has pith through which plasmodesmatal connection exits between the cell. The phloem parenchyma store food materials and other substances like resin, latex, and mucilage. Phloem parenchyma is absent in most of the monocotyledon. Phloem fiber, bast fibers, are made up of sclerenchymatous cell. They are gradually absent in primary phloem but are found in the secondary phloem. These are much elongated, unbranched, and have pointed needle-like apices. The cell wall of phloem fiber is quite thick. At maturity, these fibers lose their protoplasm and become dead. Phloem fiber of jute, flax, and hemp are used commercially. The first formed primary phloem consists of narrow sieve tube and is referred to protophloem, and the later formed phloem is bigger sieve tube and referred to metaphyloem. 6.2 The tissue system. We were discussing type of tissue based on the type of cell present. Let us now consider how tissue vary depending on their location in the plant body. Their structure and function would also be dependent on the location. On the basis of their structure and location, there are three types of tissue system. These are epidermal tissue system, the ground or fundamental tissue system, and the vascular or conducting tissue system. 6.2.1 Epidermal tissue system. The epidermal tissue system forms the outermost covering of the whole plant body and comprises epidermal cell, stomata, and epidermal appendage. The trichome and hairs. The epidermis is the outermost layer of the primary plant body. It is made of elongated, compactly arranged cell which form a continuous layer. Epidermis is usually single layer. Epidermal cells are parenchymatous with a small amount of cytoplasm lining the cell walls and large vacuole. The outside of the epidermis is often covered with a waxy thick layer called cuticle, which prevents the loss of water. Cuticle is absent in root. Stomata are structure present in the epidermis of leaf. Stomata regulate the process of transpiration and gaseous exchange. Each stomata is composed of two bean-shaped cells known as guard cell, which enclose stomatal pore. In grasses, the guard cells are dumbbell-shaped. The outer wall of guard cell away from the stomatal pore are thin and inner wall toward the stomatal pore are highly thickened. The guard cell possesses chloroplast and regulate the opening and closing of stomata. Sometimes a few epidermal cells in the vicinity of the guard cell become specialized in their shape and size are known as subsidiary cell. The stomatal aperture, guard cell, and surrounding the subsidiary cell are together called stomatal apparatus. The cell of epidermis bear a number of hairs. The root hairs are a unicellular elongation of the epidermal cell and help absorb water and mineral from the soil. On the stem, the epidermal hairs are called trichome. The trichome in the shoot system are usually multicellular. They may be branched or unbranched and soft or stiff. They may even be secretory. The trichome help in preventing water loss due to transpiration. 6.2.2 The ground tissue system. All tissue except epidermis and vascular bundle constitute the ground tissue. It consists of simple tissue such as parenchyma, collenchyma, sclerenchyma. Parenchymatous cells are usually present in cortex, pericycle, pith, and medullary rays in the primary stem and roots. In the leaves, the ground tissue consists of thin-walled chloroplast containing cell and is called mesophyll. 6.2.3 The vascular tissue system. The vascular system consists of complex tissues, the phloem and xylem. The xylem and phloem together constitute vascular bundle. In dicotyledonous stems, cambium is present between phloem and xylem. Such vascular bundle, because of the presence of cambium, possesses the ability to form secondary xylem and phloem tissue and hence called open vascular bundle. In monocotyledonous, the vascular bundle have no cambium present in them, hence since they do not form the secondary tissue, they are referred to as closed. When xylem and phloem within the vascular bundles are arranged in alternate manner along the different radii, the arrangement is called radial, such as in roots. In conjoint type of vascular bundle, the xylem and phloem are jointly situated along the same radius of vascular bundle. Such vascular bundles are common in stems and leaves. The conjoint vascular bundle usually have the phloem located only on the outer side of xylem. 6.3 Anatomy of dicotyledonous and monocotyledonous plants. For a better understanding of tissue, organization of roots, stem, leaves, it is convenient to study the transverse section of the mature zone of this organ. 6.3.1 Dicotyledonous root. Look at figure 6.6a. It shows the transverse section of the sunflower root The internal tissue organization is as follows. The outermost layer of is epiblema. Many of the cells of epiblema protrude in the form of unicellular root hair. The cortex consists of several layers of thin-walled parenchyma cell with intercellular space. The innermost layer of cortex is called endodermis. It comprises a single layer of barrel-shaped cell without any intercellular space. The tangential as well as radial wall of the endodermal cell have deposition of water impermeable waxy material suberin in the form of Casparian stress. Next to epidermis lie a few layers of thick-walled parenchymatous cell referred to as pericycle. Initiation of lateral root and vascular cambium during the secondary growth take place in this cell. The pith is small or inconspicuous. The parenchymatous cell which lie between the xylem and the phloem are called conjunctive tissue. There are usually 2 to 4 xylem and phloem patches. Later, a cambium ring develops between the xylem and phloem. All tissue on the inner side of the endodermis, such as pericycle, vascular bundle, and pith, constitute the stele. 6.3.2 Monocotyledonous root. The anatomy of the monocot root is similar to the dicot root in many respects. It has epidermis, the cortex, endodermis, pericycle, vascular bundles, and pith. As compared to the dicot root, which have fewer xylem bundles, there are usually more than 6 polyarch xylem bundles in the monocot root. Pith is large and well developed. Monocotyledonous root do not undergo any secondary growth. 6.3.3 Dicotyledonous stem. The transverse section of the typical young dicotyledonous stem shows that the epidermis is the outermost protective layer of the stem. Covered with thin layer of cuticle, it may bear trichomes and few stomata. The cell arranged in multiple layers between the epidermis and the pericycle constitute the cortex. It consists of 3 subzones. The outer hypodermis consists of a few layers of collenchymatous cell just below the epidermis, which provide mechanical strength to the young stem. Cortical layer below hypodermis consists of rounded, thin-walled parenchymatous cell with conspicuous intracellular space. The innermost layer of the cortex is called endodermis. The cells of the endodermis are rich in starch grain and layer is also referred to as starch sheath. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. In between the vascular bundle, there are few layers of radially placed parenchymatous cell which constitute medullary ray. A large number of vascular bundles are arranged in a ring. The ring arrangement of vascular bundle is a characteristic of dicot stem. Each vascular bundle is co-joint, open, and with indark protoxylem. A large number of rounded parenchymatous cells with a large intercellular space which occupy the central portion of the stem constitute the pith. 6.3.4 Monocotyledonous stem. The monocot stem has a sclerenchymatous hypodermis, a large number of scattered vascular bundles, each surrounded by the sclerenchymatous bundle sheath, and large conspicuous parenchymatous ground tissue. Vascular bundles are co-joint and closed. Peripheral vascular bundles are generally smaller than the central located ones. The phloem parenchyma is absent and water-containing cavity are present within the vascular bundle. 6.3.5 Dorsiventral dicotyledonous leaf. The vertical section of dorsiventral leaf through the lamina shows 3 main parts, namely epidermis, mesophyll, and vascular system. The epidermis, which covers both the upper surface adaxial epidermis and lower surface abaxial epidermis of the leaf, has conspicuous cuticle. The abaxial epidermis generally bear more stomata than the adaxial epidermis. The latter may even lack stomata. The tissue between the upper and lower epidermis is called mesophyll. Mesophyll, which possesses chloroplast and carry out photosynthesis, is made up of parenchyma. It has 2 types of cells, that palisade parenchyma and spongy parenchyma. The adaxially placed palisade parenchyma is made up of elongated cells which are arranged vertically and parallel to each other. The oval or round and loosely arranged spongy parenchyma is situated below the palisade cell and extends to the lower epidermis. There are numerous large space and air cavity between these cells. Vascular system include vascular bundle, which can be seen in the vein and midrib. The size of the vascular bundle are dependent on the size of the vein. The vein vary in thickness in the reticulate venation of dicot leaf. The vascular bundle are surrounded by layer of thick-walled bundle sheath cell. Look at figure 6.8 and find the position of xylem in the vascular bundle. Isobilateral monocotyledonous leaf. The anatomy of isobilateral leaf is similar to that of the dorsoventral leaf in the many way. It shows the following characteristic difference. In an isobilateral leaf, the stomata are present on both the surface of epidermis, and the mesophyll is not differentiated into palisade and spongy parenchyma. In grasses, certain adaxial epidermis cells along the vein modify themselves into large, empty, colorless cells. These are called bulliform cells. When the bulliform cells in the leaves have absorbed water and are turgid, the leaf surface is exposed. When they are flaccid due to water stress, they make the leaf curl inward to minimize water loss. The parallel venation in monocot leaf is reflected in the near similar size of vascular bundle except in main vein, as seen in vertical section of the leaf. 6.4 Secondary growth. The growth of the root and stem in length with the help of apical meristem is called primary growth. Apart from primary growth, most dicotyledonous plants exhibit an increase in girth. This increase is called secondary growth. The tissue involved in secondary growth are two types: lateral meristem, vascular cambium, and core cambium. 6.4.1 Vascular cambium. The meristematic layer that responsible for cutting off vascular tissue, xylem and phloem, is called vascular cambium. In the young stem, it is present in patches as a single layer between the xylem and phloem. Later, it forms complete ring. 6.4.1.1 Formation of cambial ring. In dicot stem, the cell of cambium present between primary xylem and primary phloem is the intrafascicular cambium. The cell of medullary ray adjoining this intrafascicular cambium become meristematic and form the intrafascicular cambium. Thus, a continuous ring of cambium is formed. 6.4.1.2 Activity of cambial ring. The cambial ring become active and begin to cut off new cell, both toward the inner and outer side. The cell cut off toward pith mature into secondary xylem, and the cell cut off toward periphery mature into secondary phloem. The cambium is generally more active on inner side than on the outer side. As a result, the amount of secondary xylem produced is more than secondary phloem and soon form a compact mass. The primary and secondary phloem get gradually crushed due to the continued formation of and accumulation of secondary xylem. The primary xylem, however, remain more or less intact in or around the center. At some places, the cambium form a narrow band of parenchyma, which passes through the secondary xylem and the secondary phloem in the radial direction. These are the secondary medullary ray. 6.4.1.3 Springwood and autumn wood. The activity of cambium is under the control of many physiological and environmental factors. The temperate regions, the climatic conditions are not uniform through the year. In the spring season, cambium is very active and produce a large number of xylary elements having vessels with wider cavities. The wood formed during this season is called spring wood or early wood. In winter, the cambium is less active and form the fewer xylary elements that have narrow vessel, and this wood is called autumn woods or late wood. The spring wood is lighter in colors and has lower density, whereas the autumn woods is darker and has a higher density. The two kinds of woods that appear as alternate concentric ring constitute an annual ring. Annual ring seen in a cut stem give an estimate of the age of the tree. 6.4.1.4 Heartwood and sapwood. In old trees, the greater part of secondary xylem is dark brown due to deposition of organic compound like tannin, resin, oils, gum, aromatic substance, and essential oil in the central or innermost layer of the stem. This substance make it hard, durable, or resistant to the attack of microorganisms and insects. This region comprises dead element with highly lignified wall and is called heartwood. The heartwood does not conduct water, but it gives mechanical support to the stem. The periphery region of the secondary xylem is lighter in color and is known as sapwood. It is involved in the conduction of waters and mineral from root to leaf. 6.4.2 Core cambium. As the stem continues to increase in girth due to the activity of vascular cambium, the outer cortical and epidermis layer get broken and needed to be replaced to provide a new protective cell layer. Hence, sooner or later, another meristematic tissue called core cambium or phellogen develops. Usually in cortex region, phellogen is couple of layer thick. It is made of narrow, thin-walled and early rectangular cells. Phellogen cut off cell on both sides. The outer cell differentiate into cork or phloem, while the inner cell differentiate into secondary cortex or phelloderm. The cork is impervious to water due to sovereign deposition in the cell wall. The cell of secondary cortex are parenchymatous. Phellogen, phloem, and phelloderm are collectively known as periderm. Due to activity of the core cambium, pressure build up on the remaining layer peripheral to phellogen, and ultimately this layer die and slough off. Bark is a non-technical term that refers to all tissue exterior to the vascular cambium, therefore including secondary phloem. Bark refers to the number of tissue types with periderm and secondary phloem. Bark is formed early in the seasons is called early or soft bark. Toward the end of season, late or hard bark is formed. At a certain region, the phellogen cut off closely arranged parenchymatous cell on the outer side instead of cork cells. These parenchymatous cells soon rupture the epidermis, forming a lens-shaped opening called lenticles. Lenticles permit the exchange of gases between the outer atmosphere and internal tissue of stem. These occur in most woody trees. 6.4.3 Secondary growth in root. In the dicot root, the vascular cambium is completely secondary in origin. It originates from the tissue located just below the phloem bundle, a portion of pericycle tissue above the protoxylem, forming a complete and continuous wavy ring, which later becomes circular. Further events are similar to those already described above for a dicotyledon stem.